Okay, so here we are with the model of the human digestive system. So we're going to start and kind of follow all of the different digestive processes so that we can see kind of what's happening where and review as much of the anatomy as we can with this model as well. Okay, so we are beginning at the mouth. So the only place where ingestion can occur is here at the mouth. And the mouth is one of the alimentary canal organs, but it has several accessory organs that help it. You can see the teeth here that are very important to mechanical digestion. You can also see the tongue here, which is important to both propulsion and mechanical digestion. Okay, so we would ingest the food here and then start mechanical digestion here. And as we're mixing in the saliva, and you can't see any of the salivary glands here, which would be the other accessory organ that you have here, as you're chewing, you're mixing in the saliva, so you're actually going to begin the digestion of carbohydrates here as well. Okay, so you're using salivary amylase to start breaking down the complex carbohydrates like starch into smaller subunits. So then you're going to swallow the food material. It will move into the pharynx, another part of the alimentary canal, then into the esophagus. Okay, and we can follow the esophagus all the way down. And from the esophagus we move into the stomach. Okay, so we're going to continue chemically digesting the carbohydrates in the esophagus, even though we're really not adding anything new as a secretion. So the esophagus is just kind of a bystander, really, as this chemical digestion is occurring. It's just allowing the chemical digestion that started in the mouth to continue as the bolus is being pushed through. Okay, you can also see that the lumen of the esophagus is collapsed unless there's food being pushed through. It kind of looks like there might be food being pushed through right here because it's a little swollen. Okay, so once we get into the stomach, let's kind of review some of the structures of the stomach as we're here. This would be the gastroesophageal sphincter area here. It's also known as the lower esophageal sphincter or the cardiac sphincter, okay, because this is the cardia region of the stomach that is immediately next to this opening. The dome-shaped area of the stomach is the fundus. The main section of the stomach here would be the body, and the section close to the outlet of the stomach would be the pylorus or pyloric region. Okay, and the other sphincter that we have here is the pyloric sphincter. It doesn't look as impressive here as it does on one of the other models that we have. You can see the internal folds associated with the mucosa and submucosa, the rugae. You can also see the curvatures here. We have the lesser curvature here, lesser next to liver. Okay, and the lesser omentum should be here between the lesser curvature and the liver. The larger curvature is the greater curvature. Okay, and the greater omentum is going to hang from this curvature. Okay, you can see a lot of muscle here because we are going to complete most of our mechanical digestion here. We're also going to be beginning the chemical digestion of protein from the pepsinogen that's secreted by the chief cells of the gastric glands here in the epithelium. Okay, or in the, I should probably say in the mucosa layer. Okay, and the pepsinogen's converted into pepsin, and that starts our protein digestion. Okay, so after some time in the stomach, we're going to start to allow material to drip into the small intestine through the pyloric sphincter. And the first section of the small intestine we're going to get to is the duodenum. You know, the duodenum is really the only section of the small intestine that has to stay stationary most of the time, so it's a retroperitoneal organ. And it has to stay stationary because it has several important accessory organs that are attached to it by ducts. The accessory organs that are attached to it are the liver, the gallbladder, and the pancreas. Okay. Now the liver we talked about in lab, and in the histological section you'll hear a discussion of the liver lobule. One of the jobs of the hepatocytes is to produce bile, and bile is an important solution that is added to the chyme as it enters the duodenum, because we're going to start digesting lipids in the duodenum. Okay, and in order for digestion of lipids to occur effectively, you have to add bile, which will emulsify the lipids or break them up into lots of tiny droplets. Okay, and that increases the surface area to make the chemical digestion more efficient. Okay, so the liver produces bile. There's a series of ducts, the hepatic ducts, leading to the bile duct, which you can see here in green, that would allow the bile to reach the food material in the lumen of the duodenum. But when we're not actively secreting bile, it backs up through the cystic duct into the gallbladder to be stored and concentrated. Okay, now in addition to the emulsification, we're doing a lot of chemical digestion. We're really finishing all of the chemical digestion in the small intestine, and the pancreas is going to help us with that, because the pancreas produces the enzymes that we need to make a big completion of that chemical digestion for really every type of organic molecule. It secretes pancreatic amylase to help with starch digestion. It secretes several protein digesting enzymes, including trypsin, and it secretes lipase, pancreatic lipase, to help with lipid digestion. Okay, so we'll break down all of these organic molecules further, and then the last bit of chemical digestion will be performed by the cells on the villi. The actual simple columnar epithelial cells will finish the chemical digestion. At that point, all of the organic molecules have been reduced to their simplest form, and they're ready to be absorbed. Okay, so in addition to doing chemical digestion here, we're also doing absorption. Okay, and you can see that we have lots of folds here on the internal surface of the small intestine. These are the circular folds. This is another way that we increase surface area, just like the villi, the finger-like projections of the mucosa, and the microvilli on the surface of the individual epithelial cells. Okay, so from the duodenum, the food material is going to move into the jejunum. It's kind of hard to see where that is here. I'd say this is probably a segment of it here. And then we kind of get a little mixed up here. Not sure where we are, but eventually we get down to here. And this section has to be the ileum because it's connecting to the large intestine. Okay, and the ileum is the distal section of the small intestine. Okay, and you can still see those circular folds on the internal surface of the ileum. Okay, and then to control movement of material from the ileum into the large intestine, we have this structure, which is the ileocecal valve. Okay, this also controls backflow of material from the large intestine into the small intestine, which is really important since there are bacteria that live in the large intestine. Okay, so I'm going to stick this piece back on so we have all of our large intestine here. Okay, so here, this a kind of dead end area or blind-ended pouch, as it's described in your textbook, associated with the large intestine is the cecum. And attached to the cecum in humans, we have this little worm-like appendage, the vermiform appendix. Okay. Now, as we move in this direction, we have the ascending colon. And then we have the right colic flexure where we have this turn. We have the transverse colon. And then we have another turn here, which is the left colic flexure. We have the descending colon. And then in behind the small intestine, we'd have the sigmoid colon. Okay, and this leads us to the rectum. Okay, and in the large intestine, we do propulsion, a little bit of absorption, some vitamins, minerals, water. No chemical digestion is happening here. We're going to move the food residue along, and anything that makes it to the rectum will be defecated, and that's the end of the digestive process.